Okay, our next speaker is Lynette Hirschman, who's at MITRE, and she's going to talk to us about the cost of curation.
So, first of all, I wanted to thank the organizers for inviting me, and I have found this extremely useful. And in fact, I will be able to go through some of these things fairly fast because a lot of these points have already been made. I'm going to have to. Is this what I'm supposed to be using?
No, the
keyboard. Okay, better. Yeah. Okay. So here's a quick outline. I'm going to talk about curation versus annotation. And this is actually important because we've heard a number of people talking about some of the different use cases for annotation, but I really want to focus on the larger issue of curation. Talk a little bit about the dimensions of cost, although a lot of these things have already come up. Look at some technology insertion points, and I'm going to give you a very truncated version of some work we've been doing at MITRE recently on crowdsourcing some biological relation annotations, and then tell you what my conclusions are, although the conclusions are very inconclusive. I will warn you in advance because I'm still in the process of gathering data for all this. So very quickly, I wanted to just try to lay out some definitions. So curation, and I'm really going to specifically talk about biocuration, is really the capture of computable information from the literature in a biological database. It's an artifact that captures this, and it's used by biologists and also, the biologists are humans, and by bioinformatics pipelines, which are, of course, computer programs. So that's what I mean by biocuration, and when I'm going to use the term curation for short. Then there's all the work on annotation that we've been hearing a lot about. It's a little bit confusing because there is, for example, in biology, the notion of gene annotation, which is identification of the location of genes and regulatory regions in a genome and their functions. And it does involve some notion of marking up of what you're marking up, which is, of course, DNA sequences and not text. But I'm really going to focus, again, on linguistic annotation, which is really the identification of linguistic structures in free text. And all of you know all this. And, of course, it can include a whole variety of things, including syntax and semantics, although linguistic semantics as opposed to, well, which overlaps in interesting ways with sort of domain expert semantics and, of course, discourse relations. So that's the kind of context. Again, several of the speakers in the past have, and earlier today have talked about biomedical curation, including Burr, who started off talking about, and I think 3 different talks have used this figure that we all got from the same place, which is the annual database issue of nucleic acids research, which every year counts the number of biological databases that are around. And so 2011 it was 380 and growing. There are, of course, very important uncurated databases in the biology world. In particular, GenBank is probably the best known of these. And it contains DNA sequences for genes, and it is done by researchers depositing the data in that database. And the reason that they do it is that they are required to do it in order to publish their articles. So you have to have an accession number to show that you've deposited something in GenBank in order to publish your paper. And that was actually enforced, it has been enforced by the publishers, which is kind of interesting. But, of course, there are many, many curated databases. Uniprot, there are a lot of model organism databases about the genomes of individual organisms like fly, mouse, yeast, worm, lots of other ones. Protein interaction, metabolic pathways. These were the early ones, for example, Flybase, which we worked with a while ago, were really designed for use by biologists, so they had a centralized resource to go and find out roughly what had been published about their particular organism. But this has, over time, morphed into a very critical resource that feeds computational downstream analyses, so it needs to be computable. And that has caused a fair amount of reorganization in these curated databases. And the last thing I would say is biocuration is a growth industry, although funding restrictions may affect that. And it is also a career path these days. There is the International Society of Biocurators, which now is up to, I don't know, 300 curators. And they have annual conferences, or pretty much annual conferences. So, and there have been a lot of publications about what to do about big data and how to keep up with the need for curation. And this is just from a little excerpt from an article that appeared in Nature in actually 2008, listing some of the roles of biocurators. And their roles include extracting knowledge from published papers. So what a PhD biologist, what a curator does, who is often a PhD biologist, is read a paper and then extract that information into a database. And I'll give you a quick example in just a moment. They not only need to extract this knowledge, they need to connect the information between databases. So you see a gene mentioned, and you need to connect that to its unique identifier in some commonly used resource, like EntreGene. You need to inspect and correct automatic predictions. So that's another role. And critical part, you need to develop and manage structured, controlled vocabularies, and that is, of course, for downstream computational purposes. So that's just a quick idea of the burdens that we expect biocurators to take up. Here's a quick example of curation from Flybase. And what you see circled in red there is the particular protein, or actually polypeptide technically, which has a name. And this is the page about that particular protein. And the curator is reading an article, and here's actually a legend from a figure, which you can't possibly read, but it says whole mount tissue staining using an affinity purified anti-PHM antibody in the CNS, blah, 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 central nervous system. And what they need to do is use that information to say the assay that was used as experimental evidence for this finding was immunolocalization. So it requires a lot of expertise and detailed biological knowledge. And in particular, one of the functions of the biocurator here is to translate these very complex findings into a controlled vocabulary. And by and large, we've been working with biocurators and running Biocreative for a number of years now, since I think 2004. The further away the controlled vocabulary is from how it's actually stated in the literature, the harder the task cognitively. So this is a particularly nasty example. So one of the things that we can do when we're thinking about the curation process is look at the stages in the curation pipeline. And actually, Charles Elkin's talked about at least some of these stages. And the first stage is typically triage, is to find the papers that you actually want to look at that are relevant to your domain of expertise or to the database you're curating. And of course, each database has slightly different criteria for what is going to be relevant to them. The second stage is having found and ranked those papers. You want to be able to do indexing, so list the entities, the relevant biological entities in that document. But we're still talking at the document level here. And then the third step typically is actually doing the extraction or detailed curation, which is the extracting the findings and usually the experimental evidence from the full text of the papers. And I emphasize here full text because the corpora that are widely available for use in various kinds of natural language processing experiments and information retrieval experiments are actually often only the abstract of those papers, but a lot of the experimental evidence is in the full text. So those are the stages in the curation pipeline. I just want to give you one quick example of what I see as a predominantly linguistic annotation task versus a curation task, and that's gene mention versus gene normalization. So here is, actually it's a piece of an abstract, and the gene mention task here is to annotate every gene that is mentioned in the abstract. And I've color-coded them. There happen to be 2 genes mentioned here. They occur in somewhat different with somewhat different synonyms. For example, here we have the mention of esterase 6, and then we have EST6 is actually a synonym of that. And there's another, one mention of one other gene, leucine aminopeptidase, or actually these are proteins, but are gene products. But anyway, that's the mention task. Complete, get all the mentions. The gene normalization task, by contrast, just says, here's a list of the genes using some set of unique identifiers of the genes that occur in this case, abstract. So this was a Flybase, so we were using Flybase gene identifiers, and so there are two, and you just get these gene identifiers, and that's it. So you're sort of collapsing the redundancy. So obviously the gene normalization task is much more succinct in some sense. You're making fewer annotations, and they are at the document, in this case the abstract level. This was just the actual list of, or partial list of the synonyms, and you can see some of the problems with biomedical nomenclature because, for example, there are lots of different variants of these synonyms, including, ironically, EST5, which seems to be, has been on at least one occasion used as a synonym for EST6. Anyway, so the nomenclature is obviously not in great shape. What is the need for curation? Most of my examples so far have been from the biomedical area, and these are critical resources for omics, that's genomics, proteomics, metabolomics, but also translational medicine. And in particular, we've been working recently with databases on genetic variation related to disease. This is the Online Mendelian Inheritance in Man or OMIM, dbGaP, which is database of genotype and phenotype. Anyway, there are a number of these different databases that are basically biomedical, but there are also some medical databases interestingly, as far as I can see, fewer than what we're seeing in the biomedical or the more biology-oriented area. But there are databases for pharmaceuticals. So this little screenshot is of Daily Med, which is a curated database that has information on drugs. There are pharmacogenomics databases, for example, PharmGKB, for gene-drug susceptibility interactions. There's clinical trial criteria in clinicaltrials.gov. And a task that is familiar to many of you in the medical area, namely cohort selection, it is in some sense analogous to a curation need, but it's usually done as a kind of one-off exercise. So there really is, if you're trying to select a cohort of patients who meet certain criteria and a control cohort, that's usually done, and you do your experiment or your analysis based on that, and then you throw the data away. I mean, there's no permanent repository of that information. So it's sort of interesting to think that, to see that there are the same kinds of things going on with clinical data, but not necessarily being preserved. And of course, the reasons having to do with security and privacy, as being discussed in another workshop, that have to do with some of this. But it's interesting that there's overall less curation activity as far as I can see in the medical domain. Who pays for this? Basically, many of the biological databases are publicly funded, and NCBI and National Library of Medicine themselves maintain a very large number of public databases. Things like the model organism databases and a number of others are also funded by grants, a lot in the US, but also the European Bioinformatics Institute, EBI, funds a lot of databases. This is just a growth of various activities at NLM that actually stopped in 2000, so it's continuing to go on its exponential curve. There are also commercial databases, and I looked up one, which is HGMD, which is the Human Genome. I can't remember now. Human Gene Mutation Database, which has people curating data from the literature, and that costs almost $2,000 a year for a single person academic license. So the value of these things and what people are willing to pay for these well curated databases is really significant. So I'm now going to delve into some dimensions of cost, and a lot of these have been already covered, and Harry did just a nice job on some of these things, like the quality requirements. What kind of precision and recall do you need? What kind of reproducibility from your annotators or curators? On the curation side, how do you go about recruiting, training curators? How many do you need? And obviously that depends on your throughput. What level of expertise do these people need? And that's a very interesting question that I'm going to touch on when I talk about some of our crowdsourcing work. What's the value of information? I've been starting to put out feelers to people in the community to get their results. And one of the things that's interesting is it's very hard to get any information on the value of the information to the person using it, and also the cost of errors. So how bad is it to put something wrong into a curated database? It's probably worse to put something wrong into a curated database of gene-disease relationships than it is to, mark a wrong part of speech, for example, just to mix two completely incomparable things. Last thing I will touch on is the issue of scalability and throughput. So one of the things about the biological curated databases is they need to be maintained, and they need to be maintained hopefully at the rate that the literature is coming out at, and that was, of course, what Charles Elkin was talking about. And, where can we put tools in to help this process? And of course, finally, these things have to be maintained, and there's a workflow. And what I would say is these things are very conditioned by the particular use case. So you have this trade-off of more data versus cheaper data versus better data. You have greater curator expertise, which may cost more, versus cheaper curation. You have a one-off annotation effort to prepare a corpus versus a more steady-state maintenance that you need in a biological database. So all of these things may affect the cost and where you want to make trade-offs in that. So again, this is just to summarize some of the salient differences between the biocuration and the linguistic annotation use cases, where biocuration is really focused on this repository. The product is database entries. It needs to be scalable, accurate, timely capture. It doesn't need to be complete. In other words, it is expected by the biologists that the database may lag a little bit behind, the latest experimental findings, and they're willing to deal with that, because having some well-curated data is better than nothing. Linguistic annotation, of course, is used to create training and evaluation sets for natural language-based applications and for research. Its lifespan is somewhat different, or maybe somewhat different. Certainly for evaluation, you have 1 or 2 cycles of evaluation with blind test data, and then it's sort of done. As a training resource, these corpora can have extended lifespans, like the linguistic tree bank, the Penn tree bank. Although I was very interested in the talk about how to use partially annotated data, in general, what people have demanded was completely annotated gold standard data, certainly for evaluation, and measured by inter-annotated agreement. We've heard lots and lots about that already. Training data, and I think Bob Carpenter pointed this out, and several other people as well. Training data can be more, if the goal is to simply produce a model, a system that does something, it can be, presumably, you can make your own trade-off between quantity and quality there. So here are, thanks to a number of people, some initial figures, and this is kind of work in progress, on the cost of various of these activities. So Martha Palmer gave me some detailed figures for tree banking and prop banking. Here are some of the details on tree banking. Let me just cut to the bottom part here, because there's a lot of data here, and I had to sit down with a spreadsheet and make sure that I tried to make sure everything added up. But the rough, actually the first point is that the initial tree banking, which started back, I can't remember, in the 1990s, Mitch Marcus estimated the cost at $1 a word for tree banking. Since then, there's been a lot of progress, and the costs have come down. What I'm interested in here is a kind of notion of steady state cost, assuming that this is an ongoing process. You've made some of the upfront, investment in the software and the workflows and the guidelines and so on. How much does it actually cost a curator to do this? And from Martha's data, it came out to be something like $3 to $5 a sentence, depending on how you counted the initial setup time, which she's talked about as $70,000 training tree bankers over a period of 6 months. If you take a short document, let's say 10 sentences, that would come out to be something in the order of $50 a document. And the throughput would be something like 250 of these short documents a month. But of course, the actual costs vary. Here's the same kind of thing on prop banking. Let me just cut down to the bottom here. It came out actually fairly comparable, $4 a sentence, $40 for a 10-sentence document. The throughput was something on the order of 1,300 sentences a month. And now I'm starting to calculate something else, which is the cost per annotation. Since Martha gave me enough data to be able to do that, it came out to be something like $1.60 per predicate, which is what's being annotated in PropBank. So one of the points to be made here is that we're talking here at the level of sentences per month. So that's sort of the scale of linguistic annotation. So here's data from Brett, and I hope I didn't garble this too much, Brett, but you'll tell me if I did. So again, cutting down to, these are based on the 2010 and 2011 I2B2 challenge data sets. The tasks were medical concepts, negation, uncertainty, relations in 2010, and co-reference for 2011, but of course the co-reference built off some of these things as well. And I think the dollar figures are taken straight out of your email, Brett, so I'm pretty sure those are right. There's $40,000 for 2010 and $38,000 for 2011. Again, I've just done something very oversimplified. I took what Brett gave me in terms of the number of annotations for these corpora and said, okay, these cost, the throughput was about 800 notes annotated in one of these, for these evaluations per roughly 12-month cycle. Is probably less than 12 months because people were doing other things. And the cost per annotation, however, interestingly, is sort of in the 25 to 50 cent per annotation range. So that was, and so these are all things that I would describe roughly as linguistic annotation, although they certainly have some semantic components that are definitely subject matter specific. Just by contrast, here is some data I got from the Comparative Toxicogenomics database, who keep very nice current statistics on all this. They do curation of full text articles, so not abstracts, not short documents, but long documents like 7, 8 page journal articles. And they do it, the entities they're interested in are genes, diseases, and chemicals, and the interactions they're interested in are these pairwise interactions between these three groups. Their annual throughput roughly is about 6,000 full-text articles curated per year. Although they did a big special project as well where they curated about 10 times that amount. They gave me some interesting results, which I'll just spend a minute on. They figured out that as part of their process, of course, the triage is finding the articles that you want to curate. They do some preliminary ranking and found that they spent only about 7% of their time rejecting papers. So it took them like 2.5 minutes to figure out a paper wasn't curatable, but when a paper was curatable, they spent 20-some minutes on it. So time per curatable paper was about 20 minutes a paper, and out of that, they typically, on average, curated about 30 interactions. They figured, therefore, the direct cost was about $10 a paper. But again, if you come down to the per interaction cost, it's about 33 cents per interaction. This is, again, these are very crude figures. We haven't figured out a good way yet of sort of comparing, these are apples, oranges, bananas, tomatoes, everything. But anyway, they're numbers. So the question is, okay, so can we decrease the cost of curation? What can be automated? Triage is an obvious example, and Charles Elkin gave a nice example of how that could be maybe speeded up. It may not be the bottleneck for at least in the CTD case, but the curators all seem to want to do this. They hate the notion that they're wasting their time. Indexing is a really obvious use, a way to reduce the costs, and automated tools could certainly help with linkage to standard nomenclature, which is something that people aren't terribly good at. And this would also apply to the case that Harry was talking about, where you had a user clicking, a number of times just to get the UMLS code or the CUI that they wanted. Extraction is, of course, the least reliable thing that we have in our NLP toolkit, but curators are definitely interested in interactive tools, especially for full text where it could help them focus in on the area where they want to do the curation. One of the other questions is, the role of a curator. How much redundancy do you want? This has to do with the inter-annotator agreement issue, and it's the value of the reduced error rate, maybe, versus the cost. Expertise is something that hasn't been talked about a lot. I'm going to hit on that in a moment. Experts are, of course, more expensive in theory, at least than non-experts. Can you get community curation, volunteer curation, or non-expert curation? And then, of course, the issue of recruiting and training annotators. So, all right, now I'm going to switch gears and tell you very fast about an experiment that we did at MITRE over the last 6 months or so. This was focused on doing crowdsourcing for curation, and it was a hybrid approach. The hypothesis was, can we combine automated pre-processing to do some of the entity extraction, present that to the human annotators, and have them do basically accelerated relation annotation. So the reasoning behind this was automated entity extraction is reasonably good. Automated relation extraction is still pretty hard in terms of performance, and crowdsourcing, and we used Amazon Mechanical Turk, enables these new models of annotation. So what we did was we had the following workflow, and in fact probably, let me go back to, we were going to automatically extract the basic elements, genes, mutations, diseases, except we didn't really get to the diseases part, automatically prepare those at the level of mentions in order to present an interface that was hopefully very usable to the person doing the annotation, and have them make very simple decisions, yes or no. Okay. Have to go very fast. And then collect and process judgments on those relations. So this is what the interface looks like. We can see that the genes and proteins are color-coded in, the gene, we are asking a very specific question. Is this gene related to this mutation? The gene is in pink, and there may be multiple mentions. The mutation is in, yellow or green, depending on how you identify colors. And they were to click yes, no, or the annotation didn't make any sense. This is the workflow, and the idea was that we did some automated processing to extract mutations and to do gene mention finding, integrated that with a document tool called the MITRE Annotation tool for visualization, presented it to the Turkers. The Turkers made their judgment, and then we did some scoring and error analysis. And we did this all with people, our collaborators from University of Maryland, Marcel Kahn, and a collaborator from Harvard as well, June Lee. So we did 50, we had a gold standard that had been independently created by the folks at UMBC, and we took 250 abstracts and just did gene mutations. These are the results from the automated pre-processing pipeline, and what you should look at down here is the recall. So the idea is we were going to pull out all the genes and all the mutations and present them in turn as the cross product of candidates to the Turkers. So if we lost something out of the recall and in the pipeline, the Turkers were never going to see it. So this was obviously a partial annotation. We lost 18% on the recall. The only thing I want to call out here is that this is not free. Somebody said, oh, they're almost free. They're not free. We paid 7 cents a judgment. The total cost for this experiment was about $670. And the other thing to mention is we used control items so that we could do some kind of intelligent aggregation where the control items are things for which we, in theory, knew the answer. The baseline was dumb baseline. If you just said no to everything, you would get 58.7%. The average of the Turkers who did 100 plus items, some people did 1 or 2 and then they quit, but the average was 76% accuracy. The best Turker did pretty well, 90.5. When we do the aggregation results using various methods, it turned out naive Bayes was the best, and that bumped it up to 84.5. We did some post hoc error analysis where we got rid of some normalization errors and some annotation drift, and that actually improved things quite a bit. 2 things to call out. The naive Bayes aggregation went up to 88.3% accuracy. The best Turker was now 94.5% accuracy, which was impressive. So what can we conclude? One Turker did the task, or we can conclude that some Turkers, and these were not particularly experts, I mean, they were whoever came across the task and stuck with it. One Turker did very well. The other Turkers, it turned out, didn't add much value. In fact, they kind of lowered the score. That suggests that you might be able to use crowdsourcing to recruit good curators. But that's not necessarily, how people are using it now. It was a very scalable effort. I didn't stop on this, but it had very low latency. From the time of task release to completion was 36 hours wall clock time. That's fast. So high throughput and good scalability. Accuracy was not great, although if we could just have the best Turker, we would have done quite well. And the cost for us, it was about 50 cents an abstract per Turker. About 20 cents a relation per Turker, but if you had to do that, and we did 5-fold Turking, that came out to be $2.70 an abstract. Not clear it helped very much, and it's not clear what a sustainable cost is for this kind of database. So I'm going to skip this. And just say quickly, what are we doing next? One of the things we're interested in is, can we do better vetting of the Turkers? That might, of course, lose in terms of the throughput because fewer people would sign up and they'd take longer. Can we use this as a good interface for expert annotators? Because we think that it turned out this interface was easy to use. The best Turker wrote back comments saying this is fun. So that was a good sign. And can we use it for harder tasks? We really started out working on gene mutation disease. Not clear, and that's illustrated here. It's a more complicated task to annotate a 3-way relationship, and certainly when I was prototyping it and trying to do it, I had some trouble with it. So anyway, that's still open question. So conclusions. So the cost for linguistic annotations, we've seen figures like $3 or $4 a sentence, $500 a patient note. For the biocuration side, we've seen like $10 to $20 for a full text article. It's a big difference here. Our gene mutation relation experiment, it was $2.50 an abstract, but that was for 5-fold Turking. Maybe if we could do it with fewer Turkers, we might get better accuracy and cheaper. What's interesting is the cost per relationship here seems to be in a much smaller range. So it's like 25 cents to $1.25. Obviously, there's a lot of variability in quality here, but it really led me to the, perhaps in hindsight, extremely obvious conclusion that if you have higher level annotations, so you're annotating fewer relationships, it's cheaper. So that is not an earth-shaking conclusion, but it does say something about whether we need to think about whether we really need this kind of very detailed linguistic annotation for all the tasks, and how we're going to be able to scale up to the patient level data that we need for real medical applications. That was one of the driving forces behind my looking at this. Anyway, there are a lot of interesting approaches to deal with increasing curator productivity, and we've heard about some of these. Texpresso, by the way, is a text mining tool that's used in the model organism databases, and at least some of their measured experiments have shown that it could increase efficiency by at least 8-fold. That's a pretty stunning number. And of course, this has been the focus of particularly the biocreative evaluations, which I'm involved in, and Jiyoung Liu is also involved in. Some of the other challenge evaluations, I2B2, of course. But we really need data at curation scale, I would say. I mean, application scale, which in this case is more like curation for the training and evaluation of these systems. And just in conclusion, I would say the biocuration community is really exploring many of these options. If we were at the end of last talk, we're talking about, in effect, is there a market for this? Is there a market for annotation tools? I would say there certainly is some market for curation tools, and they're slightly different, but highly related. And so I think that it's going to be very important to get people to work together with the curators. The publishers are also very actively involved. The ontology developers are a key part of that, and of course the research community. And with that, I just wanted to quickly mention my collaborators on the crowdsourcing experiment, my MITRE colleagues, June Lee and Emily at Harvard, and Emily Dowdy and Marcel Kahn. The curators and annotators run these large-scale efforts like Martha Palmer, Brett South, and people at CTD, Carol Mattingly and AP Davis, and then all my biocreative collaborators. As I said, Jiyoung Liu is actually here. So thank you very much.
Thanks. This was a really interesting talk, and I like, they are apples and oranges and everything, but I think you did a really nice job of sort of putting them side by side so we could really look at them. So I found it interesting that you found that the Turkers were actually quite a bit more expensive if you used them as you had, but you also sort of had future plans to work with Turkers. So I guess how do you reconcile that? Is the hope that you can drive down the cost of Turking? Is the hope that they become some sort of pilot for other changes that you then give to the curators? What's the idea?
Well, I think one of the thing that actually kicked off all this work is we realized that if we could somehow do a better job of vetting the Turkers, we might not need to use so many Turkers, and indeed using many Turkers didn't help. So we could certainly drive down the cost. If we had an oracle that told us that who the best curator was, we could have just not used the other ones, right? So that would drive the cost down by a factor of 5. And it puts it in the ballpark of good curation. And the other thing that was surprising to us was that I'm sure that the person who did this Turking had some biology expertise, but you can go out and recruit people without necessarily explicitly choosing from this pool of people who have PhDs in biology, or maybe that person did, but.
Nice presentation. It struck me as you were describing the costs that one implication of this is that we need to be very careful about what text corpora we select for annotation, because it's a huge investment.
Absolutely, absolutely.
Did you go back in and test the things that the mechanical Turkers did against reality, or did you just use the existing gold standards? Because every time I've done a mechanical Turk task and aggregated the results, it's been better than the gold standards. It's been better than anything we've seen at LDC and so forth.
We did. First of all, we had this externally provided gold standard, which had been put together for a completely different purpose by biomedical experts. when we did the first experiment, I sort of glossed all over this and talked about the post hoc analysis. We discovered that there were inconsistencies. I mean, there had been some drift in the preparation of the gold standard. So we went back and did a careful scrub of the gold standard and also corrected some normalization mistakes we had made on the preparation side. At that point, when I got and we had to just simulate the results. We didn't re-Turk things, although we were about to do that. I did a manual analysis of all the disagreements and found that there were mostly false positives. There were very few things they missed, but the false positives were things where the Turkers had made a mistake. I mean, 20% of them, 20 out of 115 had to do with some pre-processing problems, but most of them had to do with they just made a mistake.
I also had a couple recommendations for getting better Turker quality. One thing you can do is give them qualifying tests. I don't know.
Yeah,
if you've
we're
done that, but
about to do that.
That helps train them. Becky Passano was telling me, she was talking to somebody, but I can't remember who it was, who was doing exactly what you suggested, which is running a pilot study on Mechanical Turk, looking at the people who did well, and then actually contacting those people through Mechanical Turk and having the follow-on task only allow those people. One of the qualifications can be a big disjunction. It's not a very strong language that they give you for doing that.
Yeah,
we want to be able to say and or, and they won't let you actually mix those two things together. But other than that, the other thing you can do is increase the quality requirements, so you can jack the requirements that people have completed. So we've gotten a lot of success at 99% acceptance rate levels and 20,000 accepted hits for people. It seems like you're not going to get anybody. They're more expensive. Those people are actually very active Turkers, and they won't work for the same rates, but the quality is much, much higher.
Yeah, and in our case, as I said, what started me down this whole path of the cost was if we could get higher quality, we would need fewer Turkers. So there's a huge return on that investment.
On that same topic, and someone brought this up earlier, and we've seen some literature too, where if you have one really good annotator, it doesn't matter how many mediocre annotators you get, sometimes it's hard to beat that one good one. I wonder if you could have a game where you're trying to train them to match that one good annotator, and then these less good Turkers could try to learn and get better. And
yeah, one of the questions I have, though, is whether they actually will learn, or whether some people are just inherently good at this. And certainly in my experience supervising annotators way before, 20 years ago, before all this stuff, some people were good at this and other people were not, period. And there was nothing you could do about it.